Chapter 5, Other Anabolic Pathways and Integration and Regulation of Metabolic Functions. So the objectives for this topic are to define anabolic reactions, describe the biosynthesis of carbohydrates, describe the biosynthesis of lipids, describe the biosynthesis of amino acids, describe the biosynthesis of nucleotides, describe the interrelationships between catabolism and anabolism in terms of ATP and substrates, and discuss regulation of metabolic activity. So the topics that we're going to cover are carbohydrate biosynthesis, lipid biosynthesis, amino acid biosynthesis, nucleotide biosynthesis, and integration and regulation of metabolic functions. So far we've talked about how we break down carbohydrates. We've alluded to how we can make carbohydrates from biosynthesis, but not all organisms are photosynthetic. So there are other ways to make carbohydrates even if you don't have the Calvin-Benson cycle. But if you do have the Calvin-Benson cycle, you can take G3P, kind of run glycolysis backwards during the process of gluconeogenesis, but you use different enzymes, and you can make starch and cellulose. If you take off from glucose 6-phosphate, you can make glycogen, like we humans do, but microbes also do too. You can take fructose 6-phosphate and make peptidoglycan if you're a bacterium. On to the pathways that don't require the Calvin-Benson cycle. So you can take amino acids, you can break them down, turn them into oxaloacetic acid, and then run them through gluconeogenesis. You can also, depending upon what their side chains look like, start them off as G3P. You can take glycerol from fat, convert it into DHAP, run them through gluconeogenesis. You can take fatty acids, make acetyl-CoA, and also run them through gluconeogenesis. So, in other words, if you have the right enzymes, you can take an awful lot of other carbon chains and make complex carbohydrates out of them. Now on to lipid biosynthesis. There's a wide variety of lipids. There's a wide variety of pathways. We're going to stick to just the synthesis of fatty acids for this class. If you have the Calvin-Benson cycle, you can generate glyceraldehyde 3-phosphate, G3P, from the Calvin-Benson cycle, take isomerase, and make DHAP out of it, from which you can make glycerol, which goes into fats. Now, on the other side, in glycolysis, you produce pyruvic acid, from which you can make acetyl-CoA. You do the reverse of beta oxidation that we talked about in the other catabolic pathways topic, and make fatty acids. Then you take the fatty acids, slap them onto the glycerol, and you get fats. Now let's talk about amino acid biosynthesis. Amino acids are synthesized from precursor metabolites, and depending upon what kind of organisms you are, you can get them from glycolysis, you can get them from the Krebs cycle, you can get them from other amino acids, or if you're lucky enough to have the pentose phosphate pathway, you can get them from there. So, not all organisms have all of these pathways. So, different organisms have different enzymes, and they can biosynthesize different amino acids. Some organisms can synthesize all of their own amino acids. Example of that from bacteria would be E. coli. The E. coli in your gut, self-sustaining as far as amino acids are concerned. If there are amino acids floating past them, it's cheaper to import them than to make them yourself. So they are capable of importing amino acids, but if they're cut off from a particular amino acid, they can turn on biosynthetic pathways and make their own amino acids. And of course, algae can too. Being photosynthetic, autotrophs, we'll be talking about that in the next chapter. And of course plants, but plants don't fall under microbiology. Some organisms can make some of their own amino acids and can't make others. So they have essential amino acids that they must get from outside of their own cells. Mammals, including humans, are in this category. Now there are some organisms that can't make any of their own amino acids. They do not have the enzymes to do so. So an example of this would be lactobacillus. So what's a wonderful place to find all of the amino acids you could ever need? Milk. That's why they like to grow in milk, is because they have access to all of the amino acids that they could possibly need. Now, let's talk about some of the ways we can actually make amino acids. So precursor metabolites are converted to amino acids by the addition of an amino group. Some organisms can do this through the process of amination. That sounds like animation, doesn't it? But you're taking an amine from ammonia and you're putting it onto a carbon skeleton. The example here is we take oxaloacetic acid, which is from the Krebs cycle. We take ammonia and we aminate the oxaloacetic acid into aspartic acid. See? So from there, aspartic acid can go to the ribosomes where it would participate in protein biosynthesis. Now, another method of moving around an amino group that does not involve ammonia is to take an amine group from one molecule and move it to another one. In this example, we take an amino acid called glutamic acid. Suppose we've got more glutamic acid than we know what to do with, but we need some aspartic acid. So we take the amine group from here and move it on to oxaloacetic acid. The glutamic acid becomes ketogluteric acid, which is also part of the Krebs cycle, and we get aspartic acid, which can go on to the ribosome and participate in protein biosynthesis. Now, transamination is far more common than amination. On to our last macromolecule, nucleotides. So, here's a reminder of what your basic nucleotide looks like. We've got a nitrogenous base, we've got a ribose or a deoxyribose, and we have a phosphate group. The ribose is derived from ribose 5-phosphate, which came from the pentose phosphate pathway. So if you don't have the pentose phosphate pathway, you need to get this ribose from a different method. So you can get it from other organisms, etc., etc. Now, on to the nitrogenous base. We derive these from glutamine and aspartic acids, which are amino acids. We get the precursors to make them from the Krebs cycle, or the pentose phosphate pathway, or from folic acid. Now, humans, we don't have the pentose phosphate pathway. We do have the Krebs cycle, but we have to have folic acid because we don't have the enzymes to take carbon skeletons from here to here. So we've got to have folic acid. There are other microbes that require folic acid, and so we have to give them to them in the media when we grow them. Now on to the phosphate group. This one's fairly easy. We get it from ATP. So then the nucleotides, which go on to form DNA, mRNA, ribosomal RNA, transfer RNA, and so on. Now for our very last and shortest topic, integration and regulation of metabolic functions. All of these pathways need to interact together for the benefit of the cell. So some are turned off, some are turned on. Let's talk about how we integrate them and how we regulate them. First off, we can synthesize or degrade channel and transport proteins as needed. So if we're in need of a certain byproduct, certain intermediate, certain substrate from the outside of the cell, we can make more channel proteins to bring them in. Once we've got more than we know what to do with, we can degrade those channel and transport proteins. We can synthesize enzymes only as needed. Later on, when we talk about regulation, we'll talk about how E. coli only turn on the genes that code for lactose-related enzymes when they're needed, in the presence of lactose. Makes sense. Why have it if you don't have their substrates around? These enzymes are expensive. If you have two energy sources, the organism is going to catabolize the most energy efficient. Say I've put my E. coli into a broth that has amino acids and glucose. Which one is it going to use first? Well, glucose. It's the most energy efficient. Takes the least amount of energy to get the most amount of energy out. Then when the glucose runs out, then they switch to the amino acids. So they synthesize precursor metabolites only when not environmentally available. In other words, it's cheaper to import it than to make it inside of the cell. Eukaryotes isolate most enzyme within membrane-bound organelles. So you only send them the substrates when you need to have the product. We can use inhibitory and excitatory allosteric sites to control enzyme pathways. So in other words, we can turn off or turn on the enzymes whenever we need them, but still keep the enzymes there ready to go when they're needed. Feedback inhibition. This is a lovely one that cells use all the time. Once we get enough of the product, then the final product goes and inhibits the first enzyme in the pathway so that the pathway is shut down until the concentration drops and we need it again, so the inhibitory product drops out of the allosteric site and the pathway is turned back on. We regulate catabolic and anabolic pathways that use the same substrate by requiring different cofactors. An example that we already talked about is in catabolism we use NADH, in anabolism we use NADPH. That way we can control what happens when by use of those cofactors, those hydrogen carrying molecules. Okay, so here's a summary of everything we've covered. Shows how everything interacts. Isn't that a beautiful figure? I would make sure that I knew approximately what was going on in this figure. How the products of glycolysis can go on to produce energy. How the products of glycolysis can go into anabolic processes. So catabolism are in red, anabolism are in blue or green. Depends upon what your computer screen looks like. So here are the reminders of what I wanted you to learn from the topic. And remember, if you have any questions, please bring them to class. I'm more than willing to go over them again.